at the 3-letter as well as the 1-letter codes for the amino acids, and these are most important because in exams, instead of giving the total name, they just give you a 3-letter codon or a 1-letter code, and you need to figure it out how and why for the remaining part of the question. So let's look at first. So we have divided it into 1, 2, 3, 4, 5 groups, right? So first one is non-polar. So let's try to figure them out. So alanine, then we have glycine, then we have isoleucine, then we have leucine, then we have methionine. 1, 2, 3, 4, 5. Well, where is the other one? Arginine. No, not arginine. This is valine. So these 6. That is glycine, alanine, valine, leucine, isoleucine, as well as methionine. If you look at this, all of them are having whatever they are pronounced. So if you look at glycine, this is glycine, alanine. So first 3 letters, glycine, alanine, it's valine, this is leucine, this is isoleucine, and this is methionine. So we see that only the first letters are taken into consideration for the non-polar. And the second one was, though they are polar, they are having no charge. And in this comes serine, threonine, cysteine, aspargine, and glutamine. So these are represented by serine is represented by SCR, so that is again S. Threonine is THR, the first 3 letters, and it is represented by T. And cysteine is again first 3 letters and is represented directly by C. Aspargine. So it is how it is sounding, the phonetic sound, aspargine. So it goes with N. Where is N over here? Here, aspargine. And glutamine. So in the glutamine, where can I find glutamine? Where is glutamine? Yeah, glutamine is over here. So it is having a different letter, that is Q. So what do you need to memorize over here? Only aspargine is N and glutamine is Q. So these two you need to remember, and also aspargine, the phonetic sound N, and glutamine, this one. And next we see the basic amino acid. So basic amino acids means they are having positive charge, and only 3 of them are there, that is lysine, arginine, and histidine. So we'll look how they are there. So lysine is LYS, which is represented by K. So we also need to remember this, lysine is K because leucine is already given LE. So this is K. Then arginine, because already A is given to alanine, arginine gets, where is arginine? Arginine is given R because it is sounding R, so it is given R. And histidine, because no other letter is designated as histidine, H, it is given H. So here we need to remember K for lysine, R, arginine, and histidine is H. And this 3-letter codons are arginine and histidine. These are similar. Lysine also is similar. So we don't need to remember that. Only K, R, H, N, Q. And then what do we have? We have the acidic amino acids. So only 2 are there. One is aspartate and one is glutamate. Because we are writing glutamine and aspargine, for these two we write it as aspartate. That is aspartate now. ASP, aspartate. So 3-letter codons is similar. How they sound is how you write them, glutamate. And so aspartate gets another letter, that is D. Aspartate gets T and glutamate gets E. So you need to remember this also. And you also need to make sure that you don't confuse between E and Q, because glutamine is Q and glutamate is E. Should not confuse between them. And aspartate is D. Aspargine is N. How it sounds N, aspargine, so N. Aspartate, so D. And what else is left? We are left with proline, tyrosine, tryptophan. And we're also left with phenylalanine. We'll see how they're written. Proline is simple P. Proline, where is it? Proline, yeah, here. Proline. So P. Tyrosine is Y. How it is sounding? Y. Tyrosine. Tryptophan is given another letter, that is W, because we have already given T for threonine. We have given Y for tyrosine and tryptophan is W, and phenylalanine. Where is phenylalanine? It is given F. How it is sounding? F. So again we need to remember. Proline is similar, so you don't need to remember. Tyrosine, Y. Ty is giving ty. The sound is Y, so it is Y. Tryptophan is a different letter, that is W, and phenylalanine is phi. Phi sound is coming, hence you give it F. So what all do you need to remember over here? You need to remember tyrosine Y, tryptophan W, phenylalanine F, glutamate E, aspartate D, lysine K, arginine R, aspargine N, and glutamine Q. So 1, 2, 3, 4, 5, 6, 7, 8 and 9. You need to remember only 9 and rest of them are all similar. So now we have already discussed about the classification of the amino acid based upon the charge and hydrophobicity or hydrophilicity. Now let's discuss about two more classification. The first one is based on whether it is essential to the body or not essential to the body. So non-essential to the body means the body can synthesize it. That means it is made in the body through some biochemical pathways or their intermediates. So such as TCA or glycolysis intermediates. And this also means that it need not be taken through some external ways such as the diet. But whereas when we look at the essential amino acid, it means that it is essential and the body does not synthesize it. So it has to be taken from outer sources, and the outer sources is diet. Now let's talk about the third classification, that is based upon the catabolism. So catabolism refers to breaking down. So amino acids, we know the general structure, that is C alpha carbon, which is connected to the carboxylic acid, and the amino group, which is having a side chain and a hydrogen. So this, when the catabolism takes place, the COOH group leaves the body in the form of carbon dioxide, and NH3 becomes urea through some intermediate steps. And the R group and the C alpha, which is left out, it has two fates. Either it can be used in the gluconeogenesis, that is formation of glucose, or it can be also used in the formation of the ketone bodies. So in the ketone bodies, there are three forms. That is, it can either form the acetone, acetoacetone, or it can also form the beta hydroxy butyrate. So these are the three forms. So based on whether the alpha carbon becomes goes into gluconeogenesis, it is glycogenic, and whether it forms into ketone bodies, it is referred to as the ketogenic, and whether it forms both of them, it's glycogenic plus ketogenic. So this is how it is classified. Now let me give you examples of them. So when you look at this, so we all know that there are 20 amino acids, right? So 20 standard amino acids which are seen in a human body. There can be some so there can be non-standard amino acids also, like the pyrrolysine and selenocysteine, which are made from the stop codons. So pyrrolysine and selenocysteine are 21st and the 22nd codons, or sorry, 22nd and 21st amino acids. So these are non-standard amino acids, whereas these 20 which we have discussed earlier in the slide are the 20 standard amino acids. So we are only going to classify this 20 amino acids based on essentiality and glycogenic and ketogenic. So first, if we look at whether it is non-essential or essential. So first one, this is non-essential. So non-essential means, as we have seen, the body can synthesize it. So all these 8 are non-essential. I'll give you a simple trick to remember this. So this is Vim, Fill, WT. So Vim, Fill, WT, that means V for valine, I for isoleucine, M for methionine, F the phonetic sound, it's the phenylalanine, leucine. So this I is just repeated, you can cross it off. So F is phenylalanine, L is leucine, the other L is the lysine, LYS lysine, and the W is the tryptophan, and the T is the threonine. So methionine, threonine, valine, isoleucine, phenylalanine, tryptophan, leucine, and lysine. Lysine is K. So these are the non-essential amino acids, and rest all. So non-essential amino acids are 8, and essential amino acids 20 minus 8 is 12. So we have 12 amino acids are such as essential ones. So now when we look at the glycogenic and ketogenic, we see that methionine, threonine and valine, all the 3 of them are hydrophobic, are glycogenic. And if we look at both glycogenic and ketogenic, isoleucine is branched. Phenylalanine and tryptophan, these both are aromatic ones. So which means phenylalanine and tryptophan are having some ring structure. So these two are glucogenic and ketogenic, both of them. And ketogenic is only leucine and lysine, so you can remember them with double L. So these are the non-essential amino acids characterized into glycogenic, glycogenic and ketogenic. And if you see the rest all, that is the 12 amino acid, all of them, in all of them, only tyrosine. Just exclude tyrosine. So 12 minus 1, that is 11 are glycogenic, and only tyrosine. Tyrosine is Y, is both glycogenic and ketogenic, and you have no amino acid which is essential, giving ketogenic substances. So now we'll study about proteins. And how are proteins formed? Proteins are formed when amino acid combine together to form a long polypeptide chain. So in the peptide bond, so we call this peptide bond also as amide bond, which is formed because of the condensation reaction or also the dehydration reaction between two amino acids. So let's look at this. This is amino acid 1 and this is amino acid 2. So when we read an amino acid, it's from N terminal to C terminal. So we have the amino group here, that is the N terminal. N represents the N terminal and C represents the C terminal. So we always put it in N to C direction. Here also it's N to C direction. Now we see that OH of the C and H of the NH2 is getting eliminated to form water molecule. So 2 H's and 1 O. It forms a water molecule and leaves, because of which there is a condensation reaction and there is joining. This joining between C double bond O and NH is referred to as the peptide bond. And because the peptide bond formed between two amino acids, this is a dipeptide. Di refers to two amino acids which have a peptide bond in them, and a water molecule is eliminated. So there are two configurations. The first configuration is the cis configuration and the second configuration is the trans configuration. So this is again amino acid 1 and amino acid 2. This is the N terminal, this is the C terminal. N terminal, C terminal. So OH of the COOH and H of the NH2 is eliminated, and both O and H are in the same direction. We have already studied in the geometrical isomerism that cis configuration refers to the functional group being on the same side. And if the functional groups are on the opposite side, this is called as trans configuration.